in front of you is glycolysis. That's what we drew out on Wednesday. So again, we're focused on the, oh boy, that is just obnoxious. So we focused on the inputs and the outputs. Again, you would not be expected to redraw these diagrams. You should just, if given a similar diagram, you could interpret it. So a couple key things on glycolysis. It's the first step. This is where you start typically with glucose. Glycolysis literally means to split glucose. It occurs in the cytoplasm. Therefore, it requires no organelle. There's no special organelle needed, and it does not require oxygen. So all organisms, whether they carry out aerobic respiration or anaerobic respiration, will do glycolysis. And basically, we're putting in glucose, and what's coming out is we're producing ultimately 2 3-carbon products called pyruvate. And in the process of creating the pyruvate, we also create a couple ATPs. We actually make 4 ATPs, but it took 2 ATPs to get us started. So there's a net gain of 2. So 4 were made, 2 were invested, net gain of 2. And also let's move this out of the way. Also, we have oxidized the glucose and reduced 2 NADH molecules. We reduced 2 NADH molecules. Reminder, those reduced NADH molecules contain the electrons that used to be in the bonds of the sugar, and it contains some of the hydrogen that used to be in the sugar. Along with those electrons and hydrogen that the NAD is holding on to, it's holding on to some of the energy that used to be in the sugar. So keep thinking of NADH and FADH2 as electron or energy carriers. That's where the energy is. So after we looked at glycolysis, we then went to the link step. The link step will only happen if oxygen is present in the system. If oxygen is present, you can enter the link step. The other requirement is you have to have the organelle, the mitochondria. So as long as you've got oxygen and you've got the mitochondria, we can go forward. And so here was the intermediate or the link step. And really what we're doing is we're prepping the molecule to be able to enter the mitochondria. So we're going to oxidize the pyruvate. Oh my gosh. Is it flashing on your screen as well? Yeah. All right. Hopefully nobody has epilepsy. Okay. It's like a strobe light here. So in the link step, we're oxidizing the pyruvate into something called acetic acid. We then merge that acetic acid with a coenzyme called coenzyme IA to form acetyl-CoA. When it's in the form of acetyl-CoA, it's able to move through the outer membrane and the inner membrane of the mitochondria and go into the matrix, the center, the dense fluid of the mitochondria where the Krebs cycle occurs. So this is just prepping the molecule to get it into the matrix so it can go through the Krebs cycle. In the process of oxidizing the pyruvate, again, you can see if it ever stops flashing, we can remind ourselves that we produce 1 molecule of CO2. We strip one of the carbons away. Notice we've gone from a 3-carbon pyruvate to a 2-carbon acetyl-CoA. And again, we've reduced a small amount of NADH. So some of the electrons, some of the hydrogen, and some of the energy that were part of the sugar are again being picked up by the NADH. That took us to the Krebs cycle or acetic acid cycle. And again, we are, just to remind you that we are, for the Krebs cycle, citric acid cycle, we're right here in the matrix. We're in the center of the mitochondria. And really what the Krebs cycle is doing, the Krebs cycle is just a whole series of redox reactions. We're oxidizing, reducing, oxidizing, reducing, oxidizing, reducing. So every step here, I guess I'll do it here. Here we're oxidizing and reducing. Here we're oxidizing and reducing. Here we're oxidizing and reducing. And every time we oxidize and reduce, we strip off more electrons, more hydrogen, more energy, and we put it temporarily into the carrier molecules of NAD and FADH. We end up completing, the Krebs cycle completes the breakdown of the original sugar. The Krebs cycle completes the breakdown of the original sugar. There's nothing left of the sugar at this point. The only thing left of the sugar, I shouldn't say nothing's left, but there's no structure of the sugar left. All the bonds have been broken. The remnants of the sugar are all part of the NADH and FADH2. That has the hydrogen that's still part of the that was left from the sugar, and it has the electrons and energy that were in the bonds of the sugar. But the sugar at this point has physically been completely broken apart. And the key is those first three steps allowed us to break that sugar apart piece by piece, little by little, so we didn't release too much energy too quickly. It allowed us to release the energy in slow, controlled steps. So all I'm left with at this point is NADH and FADH2. CO2 has been released as a waste product, and the only thing left, physically left from the sugar, is being carried by the NADH and the FADH2. So now we're going to move to that next step. So we're going to go into something called chemiosmosis. And oxidative phosphorylation, which is what the most recent polio is about. These are both part of what happens in what we call the electron transport chain. Also sometimes just referred to as ETC, electron transport chain. So the electron transport chain, which is the physical pathway, is going to carry out two processes. The first process that will be carried out by the electron transport chain is this thing called chemiosmosis. And the second thing that will be done by the electron transport chain is oxidative phosphorylation. And so there's two phases going on here during this final step. So chemiosmosis and oxidative phosphorylations are two processes that are carried out by the electron transport chain. They're not separate from the electron transport. They are part of the electron transport chain. So we'll see in a moment. Step one, the electron transport chain is going to carry out chemiosmosis and set up that proton gradient, very similar to what we saw in photosynthesis. And the second step is going to go through oxidative phosphorylation and use, again, like in photosynthesis, it's going to use this specialized protein called ATP synthase to generate ATP, which is the whole point of the final step, is to generate ATP. Up to this point, we've only produced a total of 4 net ATP. We've only produced a total of 4 net ATP. It's not enough energy. We need more bang for the buck. And that's what's going to happen here. All right, so let's sort of just draw this out. Again, put you where we're going to be. Let's go with the mitochondria. This is our mitochondria. There's the outer membrane. Here's the inner membrane. So the electron transport chain is a series of proteins. Closer. The electron transport chain is a series of proteins that are embedded in the inner membrane. There are a series of proteins that are embedded in the inner membrane. And so we are talking about, we are working right along the membranes here. There's a whole series of them all along the membrane. So we're going to try to represent that membrane. So I'm just going to draw a small section of the mitochondria. So if I enlarge the mitochondria, this is the outer membrane. And then we've got this folded membrane. This is the cristae. Cristae is the inner membrane. And just a reminder that this is the inner membrane space. So it's like I took right here, this part of the mitochondria, and sort of blew it up, the picture up. Just looking at a small section of it. Embedded in here, okay, there will be proteins, transport integral proteins. This is going to form the electron transport chain, ETC. So there's going to be, I'm just going to add a few things in and then we'll start filling in the information. So I'm just putting some arrows in. I probably should use a different color here. Those arrows are representing redox reactions, oxidation, reduction, oxidation, reduction. So we have an OX oxidation, we have a reduction. We have OX oxidation, we have a reduction. We have oxidation, we have a reduction. Oxidation, we have a reduction. I'm going to draw the exact same thing. I'm just going to go ahead and make it a little bit bigger. So don't freak out. It's going to look exactly the same. I'm just going to make it a little bit bigger. Again, this is the electron transport chain. Over here we have a specialized protein called ATP synthase. And I suppose I should have identified in here, this is the matrix. Outer membranes right here, inner membrane spaces right here, cristae, matrix. Okay, so let's see if we can put the story together on this annoying flashing screen. So remember, all that we have left is NADH and FADH2. So the NADH and much smaller numbers, or FADH2. NADH or FADH2 is going to come into is going to move towards the electron transport chain. All the NADHs that you made during glycolysis, all the NADHs you made during the link step, all the NADHs and FADH2s that you made during the Krebs cycle, they're going to move to an electron transport chain in the vicinity. And what we are going to do is we are going to oxidize them back into their original form. We are going to oxidize NADH back into NAD+. Or we're going to take FADH2 and put it back into FADH+. So we're going to oxidize these molecules back into their original form. Oh, please stop flashing. Thank you. And these carrier molecules, the FADH plus and the NAD plus, they've done their job now. They've dropped the electrons and hydrogen off that were originally part of the glucose, and they are now going to be, they're now free to go back and repeat the same job. They're going to be recycled and reused. So these guys are recycled and reused. Now, what's happened as we oxidize the NADH or the FADH2, I'm going to follow one of the NADHs for right now. So we've oxidized the NADH. That means we've stripped it of the electrons. We've removed the electrons from the molecule. And we also have stripped it of its hydrogen. So the hydrogen has been pulled off, and the electrons have been pulled off. The electrons have been pulled off, and the hydrogen has been pulled off. Two different things are going to happen to those molecules, those components. So the NADH was carrying the electrons. Remember, wherever the electrons go, so goes the energy. Those electrons were originally part of the bond of the original sugar. So the energy that's in those electrons was originally part of the sugar. And we've got this hydrogen, which has been tagging along with it. So let's look at what's going to happen with the electrons. So what happens is we start to accumulate hydrogen in the matrix. We start to get some, as the NAD and FADH cycle through, we start to get some hydrogen into the matrix. And so and the electron, when it is removed, is in a high energy state. It's an excited electron. It's at a high energy state. And so this electron is going to get shuttled. This electron is going to get shuttled down the electron transport chain. And by the time it's done, the electrons end up down here in a lower energy state. So they go from up here in a higher energy state, and as they get shuttled down the pathway by these transport proteins, the electron drops to a lower energy state. It's dropping to a lower energy state. So if it's going from a high energy state to a lower energy state, that means we're going to have some leftover energy. We're going to have some free energy available. We're going to have some free energy available. That free energy that's being released by the electrons as they go down the electron transport chain is used to run those proteins. Those are protein pumps. These transport proteins are protein pumps, which means they're capable of carrying out active transport. And what they're going to do is these proteins are going to pump the hydrogen into the intermembrane space. And you're going to start to accumulate hydrogen in this intermembrane space. We are building up that proton gradient. Exact same thing that happened during during photosynthesis. Exact same thing that happened during photosynthesis. Only in photosynthesis, we were doing it the opposite way. We were pushing hydrogen into the dome, into the stroma, and we were building up a concentration in the center of the chloroplast. Here we're moving it the opposite. We're taking from the matrix into the intermembrane space, and we're building up a concentration in the intermembrane space, but it's the same effect. So the electrons provide the energy necessary to pump the hydrogen into the intermembrane space. The electrons provide the energy necessary to push the electrons into the intermembrane space. Why do I want to put the hydrogens into the intermembrane space? I want to build up that proton gradient. That's building up potential energy. So as I put more and more hydrogen into the intermembrane space, that builds up a higher and larger concentration gradient. That also builds up that larger electrochemical gradient. When I'm done, I have left over with an electron down here that's at a lower energy state. Something has to happen to this electron. We'll come back to that in a moment, but this electron can't just hang out there. That electron has to go somewhere. Before we can do that, let's talk about that proton gradient again. Again, the hydrogen is outside the cristae, it's outside the intermembrane, it's in the intermembrane space. The hydrogen is not permeable to the cristae, just like the hydrogen was not permeable to the thylakoid in photosynthesis. So this hydrogen is getting put outside here. It's building up a gradient. So as you put more and more hydrogen in here, you get greater concentration differences. You start getting huge concentration differences. You start getting large amounts of hydrogen in the intermembrane space and very little hydrogen in the matrix. So that creates a potential energy because all that hydrogen wants to get back into the matrix where there's a lower concentration. And again, you have this electrochemical gradient, the outside, the intermembrane space. The intermembrane space is becoming more positive. In reference, the matrix is becoming more negative. So there's two forces wanting to push the hydrogen back into the matrix. Two forces. There's a concentration gradient that wants to drive hydrogen back into the matrix, and there's an electric difference, a charge difference that is also another force that is trying to push hydrogen back into the matrix. So you're building up a lot of pent-up energy. You're building up a lot of potential energy in that intermembrane space. How do we harvest that energy? Same thing we saw in photosynthesis. As this hydrogen builds up, eventually it comes across ATP synthase, and ATP synthase is how the hydrogen can start to get back into the matrix. And so the hydrogen starts to get put back in by going through ATP synthase. And remember, as you go through ATP synthase, as you're going from potential to kinetic energy, you're going going to take, that's going to provide energy to take ADP and phosphate and convert it into ATP. So we are converting ADP and phosphate into ATP as it goes through the ATP synthase. Exact same process that we saw in photosynthesis, just moves in a slightly different manner. You got a photosynthesis, you got the stroma and the thylakoid. Here you got the matrix and the cristae. So, still not quite all the way there yet. So I said there was two processes going on. Chemiosmosis. Chemiosmosis, oh boy, this is a little busy to try to show it. What color is going to show up? Let me use green. So chemiosmosis is what's going on right through here. This part is where the chemiosmosis is going on, right? We are using chemical energy to move hydrogen through protein pumps into the intermembrane space. That's the chemiosmosis. or chemically diffusing hydrogen into the intermembrane space. And then this step right here down on the bottom right, this is the oxidative phosphorylation. We're phosphorylating ADP back into ATP, phosphorylating ADP back into ATP. All right, so that's the phosphorylation. Where's the oxidative part? Well, that comes to the final piece of the puzzle. I am still left at this point. I'm getting some ATP, awesome. But I still have this used up electron that's in a low energy state. And I have this hydrogen that has been pushed back across the membrane. They can't just hang out there. You cannot have free charged ions building up in the body. Free charged ions building up in the body are going to cause a lot of damage. They're going to disrupt a lot of chemical structures, destabilize proteins, etc. So I have to account for those two electrons, and I have to account for that hydrogen. Somebody's got to take them. They are the result of an oxidation of NADH or an oxidation of FADH2. So since we oxidized NADH or FADH2, something has to get reduced. Something has to get reduced, which means something has to pick up these electrons and hydrogen. NADH lost the electrons and hydrogen. That was being oxidized. They lost the electrons and hydrogen. Somebody has to be the final acceptor of those electrons and hydrogen. And that, my friends, is where we bring in oxygen. Oxygen, thus oxidative phosphorylation. So what's the oxygen going to do? The oxygen, remember, oxygen is not stable. It's got the atomic number 8. So its first orbital has 2 electrons. Its second orbital has 6 electrons. That second orbital is not full. Remember, to be full, it needs 8. So oxygen is always looking for 2 electrons. And remember, oxygen has a very high electronegativity. So it has a strong attraction to electrons. So what's going to happen is the oxygen is going to willingly and easily accept those 2 electrons, which now gives you oxygen with a charge of -2. This is going to pick up a pair of electrons to fill its outer shell. Am I making sense there? Yay, nay? So oxygen is looking to, it's got 6 in its outer shell. It's going to pick up 2 electrons to give it 8. That means now it's got 8 protons and 10 electrons. So 8 positive charges, 10 negative charges. So it has a -2 charge of -2. Well, a negative oxygen is now going to attract a positive hydrogen. The negative oxygen is going to attract the positive hydrogen. If you've got a -2 charge, how many hydrogens will you attract? Each hydrogen has a +1 charge, so each negative oxygen is going to attract 2 hydrogens. And if you get 2 hydrogens joining with the oxygen, you, of course, form H2O. You form water, which is your final product, which happens to be what starts the whole process of photosynthesis. It comes full circle. Oxidative phosphorylation requires oxygen in order to phosphorylate ADP into ATP. This, whoops, mixed up on my markers. This is why we breathe. Right there. It's the whole reason you breathe. It's the whole reason we go through the whole process of having lungs and having hemoglobin in our blood, is we need that oxygen to be the final reduction product. Without the oxygen, we cannot pick up the electrons and the hydrogen to form water. And so if you don't have the oxygen, you can't pick up the electrons and hydrogens. Therefore, your body will not allow you to go through the link steps, the Krebs cycle, and the electron transport chain. You'll be blocked from going through those steps because you have no way to account for those electrons and hydrogen at the end of the reaction. As long as the body knows oxygen is available, it will allow you to go through aerobic respiration because the oxygen is needed to do the final reduction to form water, which is our final product. Questions at this stage? I know it's a lot to take in. If you did the POGIL, hopefully that's reinforcing it a little bit. Again, no questions because I got it. That's cool. But if you have questions, please don't hesitate, even though it keeps flashing and annoying you. So let's do some accounting, just like we've been doing. Okay? Didn't leave much room for accounting. I'm going to erase this diagram here just so I can do my accounting. So, carbon dioxide. In the electron transport chain, there is no carbon dioxide produced. It's already gone. We made 2 during the link step and 4 during Krebs cycle. That accounts for the 6 carbons that are in sugar. So there's no carbon left by the time we get here. NADH. We're not making NADH, but we're harvesting the NADH. So how much NADH is going to come to the electron transport chain for each sugar? So we got to go back and look. We're just going to scroll back. So go back to glycolysis. There's 2 NADHs from glycolysis. There is 2 from the link step, so 2 plus 2 is 4. And there are 6 from the Krebs cycle. So 2 plus 2 is 4, plus 6 is going to be a total of 10 NADHs are going to be harvested. Turns out for every NADH, those electrons carry enough energy to drive the production of 3 ATP molecules on average. There's enough energy in the electrons for each NADH molecule to drive the production of approximately 3 ATPs. So I'm going to keep track of ATP production right here. Okay, there's nothing that comes out of this. So for every NADH, you can produce 3. That didn't show up too well. It says times 3. So 10 times 3, that means we've got 30, oops. That means we can produce 30 ATP from the NADH that was produced in the earlier reactions. So we got 2 coming from glycolysis, 2 coming from the link step, 6 coming from the Krebs cycle. That's a total of 10. Each one produces 3 ATPs on average. That means on average, we can produce about 30 ATP from all that NADH we made in the earlier steps. So there will be 10 of these for every sugar showing up. 10 of these for every sugar will be showing up. For FADH2, similar idea, we go back. We had 0 FADH2s during glycolysis. We had 0 FADH2s during the link step. But we have 2 FADH2s during the Krebs cycle. So we are going to have 2 FADH2s in total show up. I briefly mentioned it, and it probably went past you, but FADH2 is different from NADH because chemically it can hold less energy. It holds less energy. At the end of the Krebs cycle, there's not enough energy, I said, to make another NADH, but there is enough energy to reduce one molecule of FADH2. So for every FADH2, you don't create 3 ATP, you only will create 2 ATP because it literally carries less, lower energy state electron. So for every FADH2, you're going to create 2 ATP. So I'm going to put times 2. So 2 times 2 is 4. We're going to create 4 ATP from the FADH2s. Yeesh. And water, we will produce water. So you're going to oxidize 10 NADHs. You're going to oxidize 2 FADH2s. You're going to produce approximately 12 water molecules per sugar. Which, think about it, C6H12. Makes sense? H12, H water molecules. So in total, for ATP in total, we have 30 plus 4, we have 34 ATP on average that is produced by the final step. 34 ATP on average is produced by the final step. I say on average because that can vary from day to day depending on how your body's feeling, depending on what you're eating, depending on what you're doing. Sometimes you can get a little bit more than 38 per sugar. Sometimes you get less than 38 per sugar. So 38 per sugar is a ballpark estimate. 34 per sugar is a ballpark estimate. In total, it's about 38. So again, if I do some accounting, go back to glycolysis, my overall accounting, I've got a net gain of 2 ATP. 2 plus 0 is 2. I have another 2 here, so 2 plus 2 is 4. So in the final overall process, I've got 2 from the from glycolysis. I have, call that the link step. I have 0 from the link step. I have 2 from the Krebs cycle. And I have approximately 34 from the electron transport chain. So on average, a typical human is going to produce about 38 ATP per glucose. That is significantly more energy than you get from just doing glycolysis. Glycolysis only allows you to get a net gain of 2. That is all you will get if you have no oxygen. That's all anaerobic respiration will provide you is 2 ATP per glucose. If you're a single-celled organism, it works. If you're a trillion-cell organism like our cells, it doesn't work. So using the oxygen, we can increase our ATP production from 2 molecules per sugar to approximately 38 ATP per sugar. That's almost a 20-fold increase. Just by using oxygen, we have almost a 20-fold increase in ATP production. If you don't have this oxygen, if you don't have this guy right here, you do not go past glycolysis. Cannot go past glycolysis if the oxygen is not present and ready to be the final electron acceptor. This process was the major breakthrough to allowing us to get to large, complex, multicellular organisms. Without the evolution of this process, life would still be at a single cell level. And evidence indicates this is all due to endosymbiosis. This process evolved ultimately from the merging of 2 early prokaryotes. Questions at this point? Just checking my time. All right, I'm going to move back over and turn this annoying flashing board off. Okay. I will post this video and the video from the other class from Wednesday sometime today. All right, let's stop this thing from going. All right, anything that is stepping out, you can throw it in the chat or you can speak up. So I'm just trying to move something around here a second. Okay. No takers. I'm just dealing with 25 geniuses that have life completely figured out. So again, these are, sorry, let me put these headphones back in. These are, again, things you would not have to redraw. Just if you're given diagrams, could you interpret what is going on? I'm going to try something here for a second. Yeah, so it's just trying some. It's apparently the smart board that's causing us the issues. Okay. So now you can look at it without it flashing all over the place. I just couldn't draw on it without putting it on the smart board. One more chance. This is pretty much what the POGIL was all about that we had just assigned recently. Okay, let me come over here then. Okay, so this was the POGIL that you should have completed before coming to class today. Anything that we can address that you recall giving you a problem? And just giving you the opportunity to ask questions if you had any. If nothing else, you may want. I'm sorry, go ahead.
The very last question.
Very last.
Yes.
Under the extension, number 20?
Yes, sir.
Okay. So if nothing else, I'll answer number 20 in just a second. If nothing else, I suggest go back and relook at the POGIL now that we've talked about it. So some of the things that maybe didn't quite connect might connect now for you. So 20 says prokaryotic cells must have energy for cellular processes just like eukaryotic cells, yet they have no mitochondria. So which phase of cellulation would be affected by the lack of mitochondria in the cell? So if you have no mitochondria, all you can carry out is glycolysis. So a prokaryote can only do glycolysis. It cannot do the link step. It cannot do The Krebs cycle, it cannot do the electron transport chain. So it is limited to only doing glycolysis, which means the most it can do is produce 2 ATP per sugar. The link reactions in Krebs cycle occur in the cytoplasm of prokaryotes in the same way that they occur in the mitochondria of eukaryotes. However, a concentration gradient across the membrane is required. So this is in some prokaryotes. This is not all prokaryotes. However, a concentration gradient across the membrane is a requirement for the electron transport chain. Propose an alternative site for this phase of cellular respiration in prokaryotic cells. So what they're trying to get to it in B is, even if they could, which they can't, even if they could carry out the link step and the Krebs cycle, you still need an embedded set of proteins in a membrane to form the electron transport chain. And so there are no organelles in a prokaryote. So what's your only option? And really the only option where you could put this would have to be in the cellular membrane itself. Would have to be in the membrane that surrounds the cell. And then you would have to try to build up your gradient that way. The problem is you're moving, you'd be pumping hydrogen out of the cell and trying to accumulate it in the environment, which just isn't going to be practical. If that's is that making any sense?
Yes, sir. Thank you.
Okay. Sorry, I don't know why it muted me. Every time I plug in or unplug this headphone, it automatically disconnects me. Most of you are probably enjoying not hearing me. All right. So let's do this. We've been online listening for an hour straight, just about. Let's just do a 5-minute break here. So 8:32. Step away if you want from the camera, go use the bathroom, get something to drink, catch 4 minutes of sleep. We'll come back at 8:32, please. Okay, 8:32. If you're there, come back on, please. Who's sharing that? Okay, so I want to take a look at the lab investigation, this one here, and just see that we were interpreting this correctly. So we should have seen that there was two different independent variables that we were testing. One, we were looking at the type of seed. By that, I mean we're looking at germinating pea seeds that were actively growing versus non-germinating or dormant pea seeds. So that was one of the differences. And then we were looking at environmental temperature. So we had set up a set of germinating peas and non-germinating peas and exposed them to room temperature at around 25 degrees Celsius as we ran the experiment. And then we reset up the experiment and set up germinating peas and non-germinating peas at a temperature of about 10 degrees Celsius. And so our independent variables were those two things. Our dependent variable is we measured how much oxygen was being consumed in milliliters. So we were measuring oxygen consumption in milliliters. Since this is cell respiration, we needed to understand that we are consuming oxygen is one way to measure the reaction. And we talked about how we had to counteract the release of carbon dioxide, which is a product of respiration, and that's where we brought the KOH in to the experiment. So our research question would have been something along the lines of, is there a difference in the respiratory rates of germinating and non-germinating peas? And another research question would have been, does environmental temperature influence the rate of respiration in pea plants? Something to those effects. Of course, our null would have predicted in both cases temperature, and germinating or non-germinating would have made no difference. All peas will germinate at the same rate, and our alternative would have done the opposite. Any problems doing the calculation? Any problems doing the calculation? I did the first one for you. So the one that I did for you was, which one did I do for you? Germinating at 25 degrees. What did you guys get? You can throw it in the chat or just speak it. What did you get for the non-germinating? Don't wait for everybody else to do it. Either speak up or throw it in the chat. What did you get for non-germinating? We had 0.028 for germinating. All right, so I'm seeing 0.004 consistently. All right, so 0.004 makes some sense. So we can clearly see there's a difference between the non-germinating and the germinating. So I'm just going to put that in, 0.004 milliliters per minute. Just so we can compare it. Throw it in, what did you get for 10 degrees for germinating? That's what I'm seeing most people on the screen get. Okay. And then for non-germinating, that was a tough calculation. Okay. So that's one way that we could compare what's going on with that. So I can see that if I want to compare germinating versus non-germinating, it's 0.028 versus 0.0004, which is, about 7 times greater respiratory rate in a germinating plant versus a non-germinating plant. I can see that even in the cold, the germinating plant is outperforming the non-germinating plant. I can also see, if I compare temperatures, 25 degrees is about twice. Is that the number you had, or did I put it in wrong? Oops, I did forget a zero. So that doesn't look right. It's about twice the rate at 25 degrees versus it is at 10 degrees. So then you were supposed to make a graph, 2 different graphs. So I put it into a spreadsheet myself. I came up with, I don't know how well we can see that because you're looking at a smaller, but came up with put it in. So your one graph should have looked something like this. The blue line represents the germinating pea plants at 25. This is the germinating or non-germinating at, this is the germinating at 10 degrees. So this is 25 versus 10. These are the two germinating. These are the two non-germinating. So you can clearly see germinating peas have a higher respiratory rate compared to non-germinating peas. And we can see there is a difference, at least in the germinating peas. There's a significant difference in temperature. 25 degrees, doubling the performance of 10 degrees. Capiche? I don't think I forgot to make the graph on that. I would have probably done a bar graph in this case because it's a simple one-to-one comparison. And in the bar graph, I would see that germinating peas with 25 degrees would be the highest bar. And I would obviously see that non-germinating peas at 10 degrees are the lowest bar as far as reaction rate. And so I'd see the two germinating peas, I also should see the two germinating peas, regardless of temperature, are outperforming the two non-germinating peas. And again, I should see 25 degrees is outperforming 10 degrees. Yay. Then we got into the questions. This, because you didn't run the lab, might have been a little more difficult to pull some of this out. Anything you can remember on 1 through 6 that you were a little unsure of? So we should have seen in number 1, we want to equalize the P volumes because we were basing this on the equation that pressure times volume equals number of molecules times temperature. And so in order to have a fair comparison, you have to have the same volume. Otherwise, you're going to change the pressure of the system. And you're not going to get a fair comparison. So you had to have equal volumes so that you were getting an equal comparison as to, because if you had different volumes of P's in the tubes, that's going to change the pressure in the tube, which is going to cause the air to move through that tube, the colored dye to move through the tube at different rates. Am I making any sense? 2 plastic beads were used because we did not want to have different numbers of P's. Because if we just added more P's, then the respiratory rate could have changed simply because you had more individuals respiring. So by adding plastic beads, you were stabilizing the volume, but you weren't adding any more organisms that were capable of respiring. So you were keeping the number of respiring organisms consistent. Yes, no? What role is the KOH? KOH's job was to absorb the CO2, so it didn't affect the pressure and the number of gas molecules, so that we could be sure that what we were measuring was only the oxygen molecules. Again, if we did not account for the CO2, we'd have oxygen pushing in, but then we would have carbon dioxide pushing back out. And that bubble in the tube would just sort of gone back and forth, back and forth, back and forth, instead of being drawn all the way in. So by removing the carbon dioxide, that allows the colored dye to move down the tube consistently. Yay. Why was it necessary to absorb all the carbon dioxide? I just mentioned that. Respirator 3 and 6 only had beads. If we saw any change, and I I don't know if this came through. It was just a check on the room conditions. If there was any change in the respirometer with only beads, either you really messed up the experiment, or there was actually sort of a small atmospheric change taking place in the room. And so sometimes you'll get a very small change. And then what you do is you subtract that from your experimental ones. So it was a control group. And it was a control group to monitor whether there was any atmospheric changes in pressure and volume going on in the classroom. As long as it didn't have any significant changes, then you knew that your results were pretty reliable. So no, their plastic beads should not have caused the change. They can't respire. Simply a change in the room or the pressure in the room. Somebody opening up a door. If it was a day we had doors and windows opened, things of that sort can change the pressure. Any concerns here on the post-analysis? We should have been able to reject our null hypothesis. Again, you should have used the data to back it up. There was a difference between germinating and non-germinating, and there was a difference between 25 degrees and 10 degrees. So I would have rejected both null hypotheses. So temperature's a little easier to explain. Room temperature, enzymes are going to be more active. Chemical reactions are going to happen at a slightly faster rate. There's more likely to be collisions of molecules because they're moving a little bit quicker, where 10 degrees tends to slow everything down. So that one's fairly easy to explain. In the chat, how did you explain why germinating seeds would have more respiration than non-germinating seeds? Go ahead in the chat, throw something in. Why would germinating seeds have higher respiration rate than non-germinating seeds? What's causing them to use energy? I mean, if they're respiring, they're using energy. It's okay if you're wrong. Give me something. I see how this group works. You're all waiting for someone like Alan to put an answer in so you can just write the same damn thing. Alan's not any smarter than any of you. Alan's a bit of an idiot. Not really, Alan. So what are seeds doing at this point? What are seeds doing at this point? Right? They're carrying out respiration, and the reason you carry out respiration is to produce energy. So what do they need energy for? What is a seed doing? Right? So I see a couple folks threw in the word photosynthesis. Do seeds photosynthesize? Think about, where is a seed? Seed is in the ground under the soil typically. So it does not have access to sunlight. So seeds don't photosynthesize. Seeds aren't photosynthesizing yet, not till the leaves get up out of the ground. So it's not for photosynthesis, right? So what a seed is doing is a seed is growing, but we want to be a little bit more biologically sophisticated with our language. In order to grow, what has to happen? Yeah, cells have to divide. Cells have to go through mitosis. So germinating seeds are going to have a much higher respiration because they are actively dividing their cells. They are actively going through mitosis, and that's where most of the energy is going. We got to get that root out, and we got to get that shoot up and get those leaves in the air within a few days, or we're going to run out of food. We got to get those leaves up so we can convert from using the food in the seed to using the sunlight to make our own food. Okay? So germinating seeds, producing the energy because they need to go through cell division. They need to go through cell division so they can grow, particularly get the roots and the stems established so that they can become self-sufficient and independent. We're making some sense there. Yay, nay. All right. So takeaways, you should, one, have figured out how you would calculate a rate for a reaction. Two, you should understand that seeds, just like animals, can have different respiration rates depending on their activity. Germinating seeds are highly active because they are actively going through cell division. Therefore, they're using more energy. Therefore, they're doing more cell respiration. Where a non-germinating seed is dormant, it is not actively dividing and growing. And so it uses very, very little oxygen. And we should also see that temperature affects reaction rates, which should not be a surprise because all these reactions are run by enzymes. And so higher temperatures are going to make enzymes more active, and the reactions are going to take place slightly more quickly compared to a colder environment where you're going to slow those enzyme reactions down, and they're going to be less frequent. Questions? Not trying to propose that was anything super difficult, but I just want to make sure that we are seeing it the same. All right, anything else on that before I move forward? What time is it? 8:48. Yeah. I still got a half hour worth of stuff to do. All right. I wanted to try to get a little bit into fermentation and anaerobic respiration, but that's not going to happen right now. So next week we meet on Tuesday and Thursday. Thursday is going to be the unit test. That's a shorter hour, by the way, on that day. That's a half day on Wednesday, Thursday, Friday. So I'll have about an hour to do the unit test. That unit test will cover all of Unit 3. So that goes back to the enzymes, photosynthesis, and cell respiration. Little bit more emphasis on cell respiration just because we haven't had a chance to quiz on it, which I forgot to mention. Maybe I finally put it out on Remind. For whatever reason, there is no topic quiz questions available through the AP Classroom for respiration. So that's why you never saw the topic quiz. Don't know why, but there's no questions available. So that leaves us Tuesday to sort of take care of fermentation, which should not be a big problem. We can do that pretty easily. I'm pausing because the assignment I wanted to give you is also based on fermentation. All right, I'll send out a note later today. I got to think this through briefly. What I may end up doing is recording a lecture on fermentation. It honestly would be like 15 minutes, nothing super long. And I might send the recording of the lecture on fermentation and then give you this case study about fermentation to complete before coming to class on Tuesday. Okay. Any other time that you have available, you can, of course, start prepping for the test. You should be able to get back to your interactive notebooks where you should have a lot of information about what you should study. Remember, the objectives are at the front of each of those interactive notebooks. If they haven't been released back, they will be today. They should be released back, and you should have access to them. Any questions? I do have a practice FRQ or two, but I don't want to give it to you as homework because all you're going to do is go online and look up the answer and not do it. Some of you are not going to do it accurately anyway. I'd rather you do it now with me. So we'll do a couple practice FRQs on Tuesday as well before doing the test on Thursday. So I suggest go back through any notes you took or go back through the 3 POGILs and just sort of walk through things and try to put the whole story together. You need to take the time to stop and go back and now see if you can put the story together. Can you go walk us through glycolysis, the link steps, the Krebs cycle, and the electron transport chain? I don't need all the nitpicky details, but can you give me the broad, okay, in glycolysis, sugars are going in, we're making some NADH, we're producing pyruvate and a little bit of ATP. Then we take the pyruvate and we go to the link step where we're going to convert it into acetyl-CoA and we release some carbon dioxide and make a small amount of ATP and NADH. And then we go, that's about the depth you should be able to go. Am I giving you enough to go on? All right. Again, there's no need to redraw it back up, but you do need to try to see if you can link it all together in your own head. I will send something out today, no later than 4 or 5 o'clock. It won't be due until Tuesday, and it won't be super long. If nothing else, prep for the test on Thursday. Anything?
If we were to get a question wrong on the lab, do you want us to like unsubmit it and fix it?
You may. Yeah, I mean, it's always a good idea. It won't be marked late. Shoot, okay. There is a big lab we normally do with this, and I've decided I think I'm going to hold off on the big lab because it's not going to, it helps, but it's not going to make a huge difference on the test. I think I'm going to hold the big lab until we're back in person, and I think it would be better for you to carry the bigger lab out in person. So there is a lab that sort of ties together photosynthesis and cell respiration all in one activity. But we'll do that when we return, which right now is on the 19th, but we'll see if that still holds. All right, we are almost out of time, so I'm going to let you go at this point. Thank you for your attention and being here. Enjoy the weekend, and we will see everybody next Tuesday. Peace out.